about the work that we've done at Rahan Meristem. And since I came here to this conference, already the third time at least now, I think that most of you remember my previous lectures. So I'm not going to repeat the background, the material, and also the methodologies. I will just mention it very briefly. And I will concentrate on the progress that we have made in the last 2 years, and I will keep you informed on where we are today in this project. So just to remind you, our objectives were and stayed the same since we began this work. So the first objective was that we wanted to create a banana plant that is resistant to the disease. We will not compromise with tolerance or anything else, but fully resistance to the disease. That was the first point that the first directives that I got from my board of directors. The second point is that we wanted this variety to be very stable. In other words, to sustain in the field with the resistance for several, at least a few years, if not forever. The other parameter that was important to us is that this banana plant that we are producing will cater for the export industry of the bananas. So being an export banana, we wanted it to be a Cavendish banana. We did not want to work with other systems because we are a company that provides to our clients Cavendish bananas for production today. And we also wanted to stay in the framework of the logistics of Cavendish bananas, including ripening and shipping and putting the fruit in boxes and so on and so forth. And then the last point is because of intolerance in the marketplace, we did not want to use GMO tools. We are using non-GMO tools to create this variety. Okay, so my next slide, I will explain why we are working with Cavendish. Why don't we work with other banana hybrids or other banana types that exist today in the market? So we think that the Cavendish has a very good yield, excellent yield actually, and you all can testify for this. The Cavendish has very good tolerance to stress compared to other varieties that we know that grow. I'm not talking about wild banana varieties, any diploids or seed-containing varieties. Okay, and we know that the Cavendish variety, relatively speaking, is really the fastest returning variety. The cycling of the plant is very, very quick. And the bunch, we all know, it's an excellent bunch, and really today nothing can compete with it. Maybe in the future there will be a competition to the Cavendish, but today, as far as we know, and I've been following banana industry for the last 35 years, and I don't know another banana variety that can compete with the Cavendish bunch. And last, it's easy to manage. Relatively speaking, the plant is not too tall. There are many very tall varieties. The plant is manageable in terms of harvesting the fruit and preparing it for shipment. So everything here is really adjusted for the export industry. Okay, just briefly, I want to speak about the methodology that we have used and what were the steps that we took until we reached where we wanted to reach. So the first step was to choose a good genetic background, a plant that we have selected previously and we know is an excellent variety, a variety called Gal. And I think that a lot of you are familiar with our Gal variety. The second point, the second step is a method that was created by Rahan Meristem, was developed by Rahan Meristem. The phenomenon is known in biology for many years now. We did not discover the genetic mechanism, but we are making use of this genetic mechanism. And basically what we do is we use genetic elements that are in the genome, they are in the chromatin, and they are mobilized from one area of the chromatin to another area. By being mobilized to a new area, they create mutation. So you can see here in this DNA bending pattern that just represents genomic DNA, you can see that this is the mother plant before we treated it to activate these elements. And you can see that there are no bands down here. And after activation, you can see the bands that represent real mutations. These are real and stable mutations that will never be erased from the genome of these plants that was created. These mutations are created by an element that is known as a retrotransposable element. Now, I'm not going to go deeper into the genetics of this. It's a very interesting topic. There are a lot of lectures that in many occasions I participate in special groups that study retrotransposable elements. But for our purpose, these retro elements that are described here, they create mutation. And it's very interesting to know that the mutations that are created, they are never mutations that are inside a gene that expresses a protein. All of these mutations are between genes, but the area in the chromatin between genes has a very strong effect on the genes themselves, as we will see. Okay, so next step, we created these mutations and we created a very large library of mutants, very large libraries in the range of about 10,000 plants that are all mutated and they are all different slightly from one another. The next step was to screen the plants, and that's a very important step, and it's a real challenge. So we are not experts on TR4, and so we prefer to send the plants for selection to the laboratory of Professor Kema, the place that he is really the world expert on doing this task, and he performed the work because of his expertise. So we prefer to do it under his domain. The next step is we used the plants that were screened at Professor Kema's laboratory, and at this point, the person that worked with Professor Kema, he's now finished his PhD, Dr. Fernando, he called the plants that after inoculating them with the disease or with the organisms that causes the disease, he called them asymptomatic. At this point, we do not say that the plants were resistant, but we don't see symptoms on the plants. We will show you later, okay? Next step, we took the plants and multiplied them, still in the Netherlands, in a private laboratory, and all of these plants that after the multiplication, the plants were sent to a field trial in one of the most devastated disease areas in the Philippines. So the plants were screened after inoculation in the Professor Kema's laboratory. They were screened in the Philippines under very severe disease pressure. Last, after they have been screened, we did a validation experiment, and you can see here, this is already a third experiment that we did in the Philippines, and you can see the results on the left side are the controls. These are Williams, regular Williams, and you can see that not even one plant sustained without being infected. And all of the plants on the right side were after the selection in the field and multiplication of the selected plants. At this point, we want to claim that all of the plants that today are from the clones that we have chosen, they are all resistant to the disease. Why do we call them resistant and not symptomatic? Simply because of the time span. We spent 6 years straight in the field in 4 field trials, 6 years, 4 field trials, thousands and thousands of plants of these lines that showed 100% resistance. At this point, we call the plants resistant to the disease. You can see here the status of these retrotransposable elements, the same as being amplified here in the very beginning of our experiments. They are still very high, even in the field, in samples that we collected from the field. So, which shows a very stable mutation. When we started the work, we decided that it's not only reasonable to produce plants that would be disease resistant, but we wanted the plant to be, as I've said before, in the standard for the export industry, and also that the traits that are in normal cavendish plants will remain in these plants. We did not want to reduce the quality or the yield of the plants that were created. So to do this, every plant that was sent to Professor Kema's laboratory had a brother or a sister, whatever you may call it, that remained in Israel. And all of these plants, this is our greenhouse in Israel, and all of the plants in Israel were evaluated for the bunch, for their return, and all the parameters that are important to banana growers in the tropics. This is the experiment that was conducted at Professor Kema's laboratory. We are very thankful to the people that did this work, and it shows very clearly the difference between the infected plants, which was the majority, the large majority of our library of mutants, and the plants that were not infected, that either were completely clean or had a very small infection that, according to Dr. Fernando Garcia, this is a very good symptom because that shows that the pathogen tried to enter to the tissue and was blocked by the resistance. In either case, This would be the resistant plant, and this would be the plant that showed symptoms. Okay, so we move to our field trials in the Philippines, and in our first trial we had approximately 7,500 plants. No, sorry. We had about 6,000 plants, and 99.5% of the plants at this point were asymptomatic. Many of these lines did not have the agronomic traits that we wanted, but they were asymptomatic to the disease. So we had to select and select and select, mainly for agronomic traits. And you can see the results of the selection here. These are the bunches that were found from the clones that were left. The second experiment that we did was for approximately 9,000 plants, and there we had 96.9% of the plants completely resistant to the disease. 96.9% of the plants were resistant at this point. Further, we went and created an experiment where each one of the lines that we selected was in the vicinity, around that line. There were a few, sometimes 5, sometimes 10, or sometimes more control plants, and all of the control plants died, okay, and only the resistant lines remained alive. So this is just another degree of assurance that our lines are resistant to the disease. This is the next experiment, and I already showed you this photo, but this photo is an excellent photo because we say that a good photo is worth a million words. So you can see all the resistant line right next to all of the lines that were susceptible to the disease. Our last experiment, we decided to plant in a new experiment the plants in doublets. So you can see that all the plants are, in each mat, there are 2 plants. One plant is from the resistant line, and one plant is from a susceptible line. Okay, and in the susceptible we included a regular gall, a gall that is not resistant before we did the mutagenesis, or a Williams, that's what they like to grow in the Philippines, they like the Williams very much, or a 218, because in the Philippines the 218 is, according to some of the companies there, they claim that the 218 has tolerance to the disease. And here you can see many, many, many examples that the control 218 has symptoms while the gall is asymptomatic, completely does not have symptoms. The distance between the 2 plants is about 20 centimeters. So in 20 centimeters of difference between the 2 plants, you cannot say that the plants were separated by an area that is maybe infected or not infected. So this gave us a very good assurance that our system works very well. Here is another example. This is the resistant gall, and this is the Williams. Here we use the 218 that got infected, and here is the gall that remained unsymptomatic or asymptomatic, or I will call it at this point completely resistant. Here is another field, and this one again is a 218 that was selected in the Philippines, and you can see that all of the plants, actually all of the plants were infected with the disease, and very close to it, sorry. I don't know why it doesn't go back. Yeah. Okay, so next to it there is the 218 that again showed symptoms where the resistant gall was asymptomatic. Okay, these are agronomic traits, and you can see that, for example, the net bunch weight is, I'm not saying that the gall was better, but I would say there is no statistical difference between the normal gall, and the gall that was created as a mutant that is asymptomatic or resistant to the disease. And you can see the same thing with days of flowering. In general, there is really no statistical difference between the two. And you can see here also that the bunch is really exactly like a normal gall. We did not stop there, and we wanted to know what is going on in the chromatin, in the genome. So we did a complete sequence of the entire genome, something like 650 million base pairs of a gall that is resistant, and the second one, a gall that is not resistant to the disease. And interestingly, what we found was that in the resistant gall, there is a landing pad of this retrotransposable element that is exactly in an area where there is a cluster of resistant genes. And these resistant genes, I'm not going to explain too much about these, but they confer resistance in many plant pathogen interactions. Okay, so we moved from there. So we have all the genomic data. We know the differences between the gall, and there were quite a few differences, all created by these retrotransposable elements. Okay, so the next move is to go to another place in the world where the disease exists, here in Colombia. So our next trial will be in La Guajira in Colombia, and we already have the plants that are ready to go into the field in La Guajira. They have been prepared here in Colombia. And they will go back to the field for another trial in Colombia. Now, in addition to our experiments with the resistant gall, we performed another big project on another variety that is very important to you people in Colombia, the Valerie variety. And this was, after many discussions that we have with the group of Tacbeco here in Santa Marta, and they asked us at first to try and create a Valerie that would be a little bit shorter than a normal Valerie. The normal Valerie, as most of you probably know, is a very tall plant. It can reach a height of about 5 meters. And so we worked on, firstly, to create a valley that will be shorter than the normal valley, and secondly, that will have, because of the short stature, it will have more energy to give to the bunch, and the bunch weight was higher than the average bunch weight of the normal valley. These are the people, our friends from Tecbaco. You probably recognize them. Okay, so we created again the mutation on the valley, and when we created this mutation, this time we said we are going to make it a little bit easier for the people that do the screening at the University of Wageningen, and we planted 2 plants in each pot. Why 2 plants? Because we said the majority of the plants will be symptomatic, but some of the plants will be asymptomatic, so it will be easier to select the plants that are asymptomatic this way. So in most of the cases, it was a scenario like this, that both plants in the same pot were symptomatic. Of course, we discarded these lines. In some of the plants only, we saw some lines that one of the plants was symptomatic and the other one was asymptomatic, and of course we chose this one. So this was an idea that we got, and we also did the same experiment in the field where we planted 2 plants per mat. Okay, so the next step, as I've said before, is to move also this valley for a selection in the field, the same as we did in the Philippines. It will be done here in La Guajira in Colombia. Of course, in order to do this, the authorities, the nice people, our friends from ICA were not very easy on us, so they had to give us a lot of chores, and also it cost us a lot of money. So we had to test all of the plants for many, many diseases, including all the viral diseases that exist in bananas in the east part of the world. Yes, Banchito, for example, and the banana bract mosaic virus that have never been seen in the West, banana streak virus that has been seen in the West, CMV, and of course TR4, although TR4 will never be in a culture medium, in a tissue culture medium. So we had to diagnose every one of the plants against these diseases, to make sure that we do not introduce into Colombia any disease or any viral disease that exists in the eastern part of the world. Okay, so this took a very long time. Unfortunately, we spent many months of negotiations with ICA, but we are happy to say that we are over this. And now the plants have passed all the tissue culture steps, and they are after hardening, and they are ready to be planted in the field. In fact, we are speaking to the Tecbaco people for planting these plants in the field in about 3 or 4 weeks. So we will see what happens in this region, and we expect the same as it happened in the Philippines. So this is the map of the field in La Guajira, not very interesting maybe. And this is a summary of all of the levels of validation that we have performed before we came to a conclusion that the plants are resistant to the disease. So these are all the steps that we took, and all of the validation parts that we took in order to declare that the plants would be resistant. So first, In vitro, we saw that the mutagenesis worked. We saw that each one of the plants on a DNA analysis, the plants were mutants. They were real mutations. The second part was the screening that we have performed, or the people at the University of Wageningen have performed for us. So that part was done also. The next step was to do the DNA analysis and discover the QTLs, so the areas in the genome where these landing pads exist and what is the situation, which genes reside in the vicinity of these QTLs. The next step is the field trials in the Philippines that also was accomplished and was completed with success. And we are here at the last part of our work where we are going to take the plants to another region of the world to test the plants, of course, under the inspection and the observation of local authorities here in Colombia. The field trial will start in the third quarter of 2023. That's when the plants will go into the field. And of course, we will test, along with the resistance, all of the parameters, all of the agronomic parameters that are important to the growers, because we want our plants to be commercial plants and not just for publications in a journal or something like that. So we are going to test the yield, the bunch weights, the number of fingers, cycling time, fruit quality, proper ripening, grain life, and Brix value of the plant. Finally, I would like to thank all the people that participated in this project that lasted 8 years. So first of all, my team in Israel, in Rahan Meristem, the R&D researchers of Rahan Meristem. Since I took this picture, a couple of these people were replaced by other people. I want to mention Luis Alberto Restrepo from Medellin. He's also here. His two sons are sitting here, who supported financially our project. Also Tecbaco, that supported the project and are part of the project. They will be doing the screening in the field. Of course, Professor Kema and Dr. Fernando Garcia, who did the screening on both varieties in the Netherlands. And the people that helped us in the Philippines, Unifruti is the company that assisted us in the Philippines in Bukidnon, an area in the Philippines. It's up higher elevations. And Yanai is sitting here, our CEO of Meristemos Colombia. So thank you all very much for your attention.